think we are frozen here. I can click. All right, that works. Okay, so first, just to think about things in a simple but blunt way, to obtain food, predators must encounter and kill their prey. But this is not an easy task, all other variables aside, as these prey species have anti-predator strategies that can include things like physical defenses, such as camouflage and armor, as well as altering their landscape use to avoid predators in time and space. And prey acquisition is further complicated in human-dominated landscapes, as these areas host altered community structures. So some animals are pushed out while others are favored. And these areas also have things like anthropogenic food subsidies, as well as altered habitat structures. So this is just things like green space, as well as early succession from development, things like that. And then in general, human-dominated landscapes have altered prey availability and accessibility. And just an example of this in human-dominated landscapes is the human shield effect. So a lot of times prey species will seek refugia in human-dominated areas and kind of exploit carnivore avoidance of these areas to lower predation risk. And then in general, most of the time, we typically think of human disturbance negatively impacting predators. However, it doesn't have to be negative. So wolves have been documented using anthropogenic linear features such as oil pipelines to increase travel efficiency and ultimately increase their predation success. And so this is important for us to know as we are interested in documenting how mountain lions respond to different natural and anthropogenic features on the landscape in areas where they feed on black-tailed deer. And so more specifically, another example of how human features can benefit prey acquisition. So African wild dogs are social cooperative hunters, and they have been documented cornering prey that would have otherwise escaped against fences and other linear features. And so regardless of the presence or absence of human disturbance or what landscape we're focusing on, habitat structural characteristics, so things such as understory cover, canopy cover, vertical complexity of habitats is important in terms of resource acquisition and resource selection in all landscapes. And from a predator's perspective, these can be positive as the structural characteristics can reduce chase distances and ultimately benefit predation success. And they can be negative as they can reduce search efficiency, thus elongating the length of the search and the energetic ramifications involved. And so, as I mentioned, our interest is understanding mountain lion responses to natural and anthropogenic features on the landscape. And so a classic method of doing this is looking at resource selection models, and these compare landscape features used and available to wildlife, and they are fundamental tools to ecology and conservation. However, they rarely account for structural characteristics within the broad land cover types. So looking at some of these more traditional models, typically they include land cover types that are implicitly associated with cover. So these are things like forest, often broken down into more specific forest classes, as well as shrublands. So those are defined as your land cover types that would have cover. And then there's the characteristically open land cover types, such as grasslands and pasture. So throughout all of these, whether they're open or more characteristic to have cover, they're assumed to be structurally uniform. So what I mean by that is looking at these two pictures, these evergreen forests would be included in those models as having the same value to different predators and prey. And so with these original models, the selection patterns and inference made from them are correct. But it leaves us to question if leaving out structural characteristics is valid for species that require those characteristics to successfully hunt, such as mountain lions. And so mountain lions are an excellent species to better understand the spatial patterns of predators or by large carnivores relative to human disturbance, as they avoid humans, but they can persist along the urban edge. And they are stalk and ambush predators, as I mentioned. So they require specific vegetative features to enhance their predation success. And this is typically in the form of dense stalking cover. And then they're also obligate large carnivores. So they require sufficient densities of ungulate prey, and they're energetically attached to consuming large prey. And so we investigated mountain lion resource selection in California's San Francisco North Bay. And this included 3 counties, Sonoma, Napa, and Marin counties in Northern California. And this area presents a pretty unique, steep urban-rural gradient. So radiating out from the urban center of Santa Rosa that has about 200,000 people, we can analyze how resource selection changes from areas with significant human disturbance to more natural and conserved areas at all extents of the study area. And across the entirety of the region, it is predominantly private lands that range in land use from urban and suburban development to more to agriculture and more natural land. And so just looking more extensively at the North Bay, so this area does have significant amounts of human development, as there is over 333,000 private land parcels in less than 9,000 kilometers. And so for some of our individual mountain lions, they actually had over 10,000 private land parcels in a single home range. So that is a lot of fragmentation to deal with. And then this area is kind of stereotypical wine country in California. So there is extensive fencing around all these vineyards. And just with the culture of the region, it's very typical to have property boundary fencing around all of the parcels. And so, as I mentioned earlier, African wild dogs, which are cooperative coursing predators, have used anthropogenic features like fencing to positively influence their hunting success. And while this hasn't been documented in a solitary stalk and ambush predator like mountain lions, it is possible that they could take advantage of these anthropogenic features to influence their predation success. And so with that in mind, we tested various hypotheses and predictions to better understand mountain lion resource selections at locations where they fed on black-tailed deer. And so with that, we first hypothesized that ambush predators prioritize areas with stalking cover that will promote killing prey over areas with high primary productivity that may just promote encountering prey. And so more specifically, we predicted that mountain lions will select stalking cover stronger than they'll select high primary productivity. And so in this case, we used primary productivity as a proxy for black-tailed deer abundance and distribution on the landscape, as they will be tied to those kind of green areas with lots of available forage. And so we also predicted that mountain lions will exhibit the strongest selection for productive areas with stalking, we predicted that they'll exhibit the strongest selection for productive areas with stalking cover, as these areas will maximize both prey vulnerability and availability. And second, we hypothesized that predator-prey interactions involving large carnivores are influenced by human disturbance. And with that, we predicted that they will select property boundaries as they may provide advantageous opportunities to corner prey. And also we predicted that they'll avoid buildings at feeding sites, but that this avoidance is relaxed when sufficient cover is present. And so to obtain the necessary data for this analysis, we captured mountain lions across the North Bay. And during these capture events, we deployed a GPS collar that will provide their location as they move across the landscape. So as you can see from the figure on the far side, we can see those kind of GPS pings pop up across the landscape. And every now and again, they'll start pinging and clumping in the same area. And we call that a kill cluster. So it's likely that the lion made a kill and is now feeding in that location. So throughout my field work, I investigated all these different kill clusters and found mostly black-tailed deer that they were feeding on, but also other things, including livestock, smaller mesopredators, and occasionally other mountain lions. And so to understand how mountain lions were responding to natural and anthropogenic features on the landscape, we used resource selection models. And these models are a use versus availability framework. So in this case, The used locations were deer feeding sites. And it is important to note that we only use those cluster locations where we found deer as the used locations, because any other of the smaller prey, so things like raccoons and whatnot, mountain lions won't feed on them for very long, so there may not be a cluster at all. So we just weren't confident in our ability to find smaller prey. And then the available locations are systematically sampled locations throughout each individual's home range. So this is a within home range analysis representing the third order of resource selection. And so in this analysis, we included 492 locations where we documented black-tailed deer killed by 21 different mountain lions. And of those 492 kills, we found 309 in forests, 57 in shrubs, 56 in areas of sparse vegetation. So these are just areas with minimal cover that can't be classified as grasslands or forests or any of those other broad characteristics. 24 in grasslands, 23 in burned areas, 19 in development, and finally 4 in vineyards. And so here are the model results. This is a lot to digest, but I'm just going to break down a couple broad things, and then we'll kind of piece it together and get the results out of this. So as for interpreting this, the red variables are distance-based variables. So these are locations where we can look at the average distance of used locations to different habitat types and the average distance of the available locations. So we can see how they're responding and if they're selecting locations more strongly than you'd expect based on that habitat types availability across the landscape. So for the distance-based variables, negative values are selection and positive values are avoidance. And for the classification-based variables, or the blue ones, positive values are selection and negative values are avoidance. And so looking back at our first prediction that mountain lions select stalking cover more strongly than areas of high primary productivity. And we'll test this by evaluating their response to percent cover and productivity variables in the, froze up on me again here, in the plot here. So as you can see, these are both classification variables. So they are selecting primary productivity, which is NPP on the plot here, but they are exhibiting a stronger selection of percent cover. And so with this, they selected primary productivity, but at each of these locations, the average value of percent cover was 43%. And thus, we can support our first prediction that mountain lions more strongly select landscapes that will promote encountering and killing prey, rather than those that just promote encountering prey. And so looking at our second prediction that mountain lions will exhibit the strongest selection for productive areas with dense stalking cover, as these areas will maximize both prey availability and vulnerability. So we tested this with an interaction term with productivity times percent cover. And so this will just allow us to gauge variation in responses to productivity with changes in the cover. So looking at this on the plot, as you remember, they're selecting both productivity and cover, but they're exhibiting the strongest selection for areas of productivity that have high values of cover. So they're maximizing those areas where deer are going to be because there's forage availability, but where the deer is also accessible to them because they can successfully hunt in those areas with high cover values. So with that, we found support for our second prediction that availability plus vulnerability kind of maximizes hunting success for mountain lions in this landscape. And so our third prediction was that mountain lions would select property boundaries as they may provide advantageous opportunities to corner prey and increase hunting success. So we tested this by analyzing their response to fencing. So that's a distance-based variable. They are selecting fencing on the landscape. And so it's interesting to note that the used locations, so the kill sites, were on average 93 meters from the nearest fence, whereas available locations were 117 meters from the nearest fence. So this is kind of just that that distance-based variable where they're closer to fences than you'd expect based on its availability. So they are selecting those areas. And so here's an interesting video, if it'll load. This camera is actually on a fence, and you can see those two eyes in the background. That's a mountain lion, but it's actually going to chase this deer up against the fence, and we found the deer dead the next day. You may not be able to hear it well, but kind of pretty foolproof evidence that they are using these fences to some extent. So yeah, that's a pretty neat catch on that camera. And finally, our fourth prediction was that mountain lions avoid buildings at feeding sites, but that this avoidance is relaxed when sufficient cover is present. So we tested this final prediction with another interaction term, analyzing mountain lion responses to buildings with increasing values of cover. And also on this plot, we just have the response to distance to buildings because that interaction term wasn't in a highly supported model. And so also, as you can see from this, that estimate is right on the zero line. So there's really no strong avoidance or selection for buildings. And with that interaction term being in a poorly supported model, we can't really make inference from that. And so overall, our final prediction was not supported. However, mountain lions are tolerating, we're frozen again. Mountain lions are tolerating human disturbance to some extent in this landscape. And so on average, the used locations, so those kill sites, were 328 meters from the nearest building. And in general, we don't think of the North Bay as a large metropolis with huge populations of people as compared to somewhere like the greater Los Angeles area. And so from a very similar study that analyzed mountain lion selection and avoidance to different resources at kill sites, they found that mountain lions were selecting development in that distance-based context. So they were closer to development than you'd expect based on availability. But even with that selection, they were still 1,325 meters from the nearest pixel of development. So this just really highlights that there is a significant overlap with humans in the North Bay, even though we weren't able to see any selection or avoidance for buildings on the landscape. So overall in the North Bay, this research is a valuable first step in understanding mountain lion ecology in this landscape, as there is no published information on predator-prey interactions or resource selection in this landscape to date. And this research can inform local land management decisions to do things such as conserving that understory stocking cover to promote suitability for mountain lions, as well as natural predator-prey interactions. And then finally, this research could have important implications to wildfire and specifically responses to wildfires. So beginning in 2017, this area had a pretty serious string of wildfires. And in response to that, it's mandated by law that all kind of understory burn hazard vegetation is cleared within 100 feet of buildings. So that can have important implications to the value and if this area will be used by mountain lions and various species of prey. And then more broadly, we documented the value of structural characteristics in wildlife use availability analyses. And this isn't typically included in these analyses. And so we did so with these locations where mountain lions are feeding on deer. And this has also, similar things have been included in analyses with ungulate birth sites, natal den sites, and overwintering thermoregulation dens. And in these other circumstances as well, these structural characteristics prove to be very important in resource selection. And so lastly, we identified conditions where mountain lions kind of maximize prey availability versus vulnerability and where they're able to successfully encounter and kill prey. So this could have important implications for human-wildlife conflict, as it kind of could inform conditions on where keeping livestock in specific areas could minimize their vulnerability to different predators, and just things as simple as clearing vegetation around pastures to minimize the use by predators. And so with that, as you all know, these research projects don't happen without a ton of collaboration. So thanks to everyone involved, and specifically thanks to John Benson, Quentin Martins, and Dave Garcelon for all their help with this. And I'm happy to answer any questions. Yes.
How do you catch a mountain lion?
So you can do it different ways in different areas. Traditionally, one of the most common ways to do it is with hounds. So you tree the lion, and then you'll dart it, get it out of the tree, then you can do your thing, you get the collar on them. In our area, however, to use dogs, we'd have to contact like thousands of landowners just because you can't tell where the dog and lion are going to go. So oftentimes we'll put out bait like roadkill deer. We'll put it take an educated guess in the woods, put a camera on it, and kind of hope that a lion starts feeding on it. Or just with all the people on this landscape, a lot of times we'll get a call that says like, hey, I found this partially half-eaten deer kind of in the back 40 of my property. And then we'll take that and put it in a cage trap. And when the lion comes in to feed, there'll kind of be a pressure plate in the back of that cage, and then that's that. Yes.
Yeah, I'm curious about the use of trail cameras in the study.
Yeah. So it's a really important thing in this area. A lot of landowners have them just kind of curiosity of what's going on in their backyard. And it has a lot of value for us just kind of in identifying what collared individuals are where, what uncollared individuals are where. And so it can lead to potential trapping opportunities in the future. And we do use like the cellular trail cameras that will send stuff to us via text a lot for like cage trapping opportunities. So actually this picture here, if we're monitoring a cage trap, we'll know for sure that we caught a lion in the trap if we're getting pictures nonstop of lions in the trap. So for my thesis specifically, I don't have any analyses based on trail camera data, but we definitely used it a ton in the field for the capture purposes.
Are the landowners pretty receptive to?
They're really receptive in that area. And I had to spend a ton of time in contacting landowners, and usually if I was able to get in contact with them, they were cool with me doing whatever. It was difficult sometimes to just get in contact with them, but that wasn't on them, ducking calls or anything. It was just calling wrong numbers nonstop. But yeah, everyone is pretty supportive of carnivores in that area. So yeah. Yes.
How long is this study?
So the captures were started in 2016, and it's still ongoing, but the vast majority of the kill cluster data is from like 2021 to 2023. Yes.
Yeah, so I just had the question that the photograph you have used in the presentation was that taken by you or?
So yeah, most of them are, some of them are from different collaborators and whatnot, but a lot of the ones obviously that have me in them or are more like trail camera kind of pictures, those are from the field out in California. Yes.
Were lion kills associated with any particular type of lion?
Right. Yeah, so a lot of times going to these kill clusters would start with a walk through a vineyard, and it would literally, like, these rows of grapes would say like Merlot and different kinds of grapes, and then you'd have to hop over some fence because these vineyard winery corporations don't care about the wooded part of their property, so there's not even gates or anything. Then you get back in the woods and there was a mountain lion hanging out, so they're making it work. Okay, cool. Thanks, guys.
All right, thanks. Next speaker is Tanessa Morris, and she will be talking on the effect of biomass water dynamics in Cosmic Ray
neutron sensor observation, a long-term analysis of waste being the different in product production. So the next question.
Hi, I'm Tanessa, and my study focuses around cosmic ray neutron sensor observations in Nebraska. I would also like to point out my co-authors, Sophia Becker, Trenton Franz, and Andy Suker. If I can find my mouse now. So this presentation, I go over an introduction to the methods used in this study, some results and discussion, and conclusions. So an introduction. So why do we bother measuring soil moisture? So by the year 2050, global population is anticipated to reach 9 billion. This means that our current global food production, specifically in the area of grains, will need to double from what is currently being produced. So agriculture is currently the leading industrial water user, and water use is less than 50% effective in agriculture. And this can be minimized by using soil moisture sensors to determine when and how much water should be applied. So in summary, what we want to do here is maximize the blue area of the water being used by crops and minimize the other areas where there are losses. So by conserving agricultural water through soil moisture monitoring, there is more opportunity to preserve our most valuable resource and be capable of feeding the growing population. So our main goals of soil moisture monitoring are to overall maximize our outputs and minimize our inputs. The slide is frozen. We want to limit our water losses, optimize plant growth, improve our soil health, and inform producers of what is going on in their environment. So there are a few common soil moisture sensor techniques currently used. So the first is traditional hand field. You go out, you pick up some dirt in your hand and roll it around. Some pros, this is cheap and it's fast, but the problem is it's highly inaccurate with an average error minimum of about 10%, and it requires years of experience to get this poor value. The next are in situ sensors like time domain reflectometry, frequency domain reflectometry, tensiometers, and neutron probes. They're very accurate in their one spot that they measure, and they're inexpensive if you're only measuring one spot. But the problem is, to capture your field variability, you need to install many, which gets rid of your pro of it being inexpensive. And the problem with a neutron probe is that you need radiation training to operate it. The gold standard is gravimetric sampling. You go out, you dig a hole, you weigh the soil wet, dry it, and weigh it again. Its calculations are very easy, but as Sophia can also attest, this is very time and labor consuming. So, and another method is remote sensing, which is currently in development with the ESA project SMOS, NASA SMAP, and Planet Labs PSWC sensors. The data is generally easy to access and generally inexpensive, excluding Planet Labs. But the problem is, you see a 3 kilometer by 3 kilometer square. You don't see your field. So if you want to know something about your field, you have to extrapolate based on this huge spatial and temporal resolution. And the other problem is it only measures the top 5 centimeters of soil, which is not where your roots are. The mouse is very finicky, I'm sorry. And there are some newer methods being used, like the cosmic ray neutron sensor and the gamma sensor. So who uses these soil moisture sensors? In a perfect world, everyone would. But as you can see before, there are many pros and cons to using them. According to the USDA 2018 Farm and Ranch Survey, only 10 to 15% of producers in the United States use some form of soil moisture sensing and soil moisture probes. Of special note is that Nebraska is the second most adopted state in the USA, which may point to its increased productivity. And the problem we see is that adoption of these devices has not significantly changed since 1984, even with these new sensors being evolved. So how do we address this? One way is with the cosmic ray neutron sensor. Essentially, it uses epithermal neutron counts to determine soil water content. It measures at a scale of tens of hectares, and it measures tens of centimeters deep. It fills the gap between our point sensors and our remote sensing sensors. So you can get a real picture of your field. Some pros of this, it's passive, it doesn't need to create its own neutrons, it's non-invasive, It works at an intermediate scale, it's easy to transmit data, and it's insensitive to many soil properties. Thank you. Some cons is that it's currently only present in research contexts due to correction values still being required. It's $10,000 to $15,000 for one sensor, and you still need to go out and do gravimetric sampling to actually make it work. And most farmers, like, it takes my dad 3 people to open an email. He's not going to be able to do this processing. So how CRNS works, so it all starts with a supernova explosion way in outer space. This produces primary cosmic rays, which reach the Earth and interact with the atmosphere. This interaction with atmospheric particles produces secondary particles, which are known as fast neutrons. These fast neutrons penetrate soil and interact with hydrogen, primarily in the form of water. And this moderates the neutrons' energies to produce epithermal neutrons, which are counted by the Cosmic Ray Neutron Sensor. So the processing is very complicated. This is kind of an overview. You have to receive your raw counts, apply filters, and then apply the corrections you can see on the left side of this, my left, your right, on the right side of the screen there. And this looks very complicated, and it is very complicated, is kind of the problem right now. And then to calculate the actual soil moisture value, you have to use the Desilets Equation. So N0 and the Desilets Equation. This is a big, scary equation. So N0 is your corrected neutron count rate over bare dry silica soils. It relies on inputs of pore water, which is determined from soil moisture measurements in the field, lattice water, and soil organic carbon content, which are determined from a lab, and are the sources of hydrogen that cannot be removed through drying. And then our other parameters are just known correction factors for this value. So when we look at N0, N0 is supposed to be one value for your sensor. This should be a straight line throughout the season because it should be one constant value. But the problem is, consistently across all of my fields and every year, there is this dip occurring in the middle of the year. Which now we introduce the biomass water equivalent, which describes the amount of water present in biomass on the surface. In summary, it is the wet plant weight plus the dry plant weight. If we look at a BWE time series, at the start of the year, it starts really low while the corn or soybean are just emerging and growing until they reach a peak in August usually, at which point they reach senescence and lose all of their water mass in their leaves. It does not seem to be a coincidence that when the neutron count rate drops is when our biomass water equivalent increases. So how did we address this? First, how the Desilets equation works. First, you have to take your inputs of soil water content through TDR measurements or gravimetric measurements, perform weighting methods, put them through the Desilets equation as our input of pore water content to render a value of N0. So there are some correction factors that already exist. There are some first order being the most important ones, like pressure. I just learned this morning that like pressure can range your neutron count from 600 counts to 18,000 counts. So the pressure is a huge influence. The neutron intensity of where you are is also a first order. Second order is water vapor. And now we're looking at a third order correction for vegetation on the surface. So our problem with the Cosmic Ray Neutron Sensor is that we are interested in soil water, but the sensor is interested in all hydrogen. So what we inherently see as people is we can see that the water in the corn and the water in the soil are two different properties. But what the sensor sees is that this is just a pool of hydrogen. It can't differentiate between this is soil, this is corn. So we need to find a way to isolate that vegetation signal to remove it from the count so we can accurately measure our soil moisture. So if you look at our relative contributions to the hydrogen signal here, note the broken axis, pore water is our primary contributor. And how this was determined, water in millimeters, it was determined by turning everything into, if you had a bucket sitting here, how much water would be there? If you condensed all of your atmospheric water, put in a bucket, that's how much water would be there. So we can account for soil organic matter, atmospheric water, lattice water, and pore water. The problem is the biomass is still a very prevalent factor here, but we aren't accounting for it. There are some existing biomass correction factors, and this looks like a lot of information. The problem is we only have 3 studies that have actually given a number for this value. All of these papers say we need one. There are only 3 values that exist, and all but one have occurred in a forest. Inherently, looking at a tree and looking at a stalk of corn, they are 2 very different things. So the problem being, when we have a forest, we have a long growth period and it reaches a lot more wet biomass. And it does have a slight water content cycle from year to year, but the problem is row crops like corn and soybean, they have these extreme peaks that occur very fast. And it's not entirely reasonable to say something that works for a forest environment is also going to work for a row crop environment. So how do we do this? For this, we are going to investigate the relationship between N naught and BWE, referring back to those figures where the N naught line went down and the BWE line went up. So the key questions here: does a single BWE to N naught correction factor exist for all row crops? What is the nature of this relationship if there is one? And can TDR sensors be used as a reliable proxy in place of gravimetric sampling for soil moisture estimates? Some hypotheses here were that a single BWE to N naught correction factor cannot be used for all row crops. A linear relationship exists between N naught and BWE, and there will be similarities between TDR soil water content values from a sparse network and gravimetric soil water content values. So the methods used in this study: it occurs out at NREC near Mead, Nebraska. There are 3 field sites used in this study: CSP1 and CSP2 and CSP3, all by itself over there. All fields are about 800 meters across, and CSP1 and 2 have 3 TDR sensors installed, while CSP3 has 4 TDR sensors installed. So some quick field stats about each of these. So for CSP1 and CSP2, there has been a CRNS installed from 2019 to 2023. It is a 2000B sensor. It is both irrigated and rain-fed. It's no-till with 3 sensors, and it has operated a maize-soybean rotation. CSP3 has data from 2011 to 2023, and it has a CRS 1000 sensor. It's only rain-fed, no-till, with 4 TDR sensors and a maize-soybean rotation. So probably the first question is, what is the difference between these 2 sensors? So the CRS 2000 counts twice as many neutrons as the CRS 1000, minimizing our error. So for all field sites, data was collected from April 1st to November 15th to eliminate the effects of snow on the surface, because that is another hydrogen pool, and to also capture pre and post-emergence field conditions as well as post-harvest. And just to reiterate the time frame here, the CSP1 and 2 were 2019 to 2023, and CSP3 was 2011 to 2023. There is no existing study of this time frame. The longest known one is 3 years, and that doesn't actually have any numbers coming out of it. They just did the study. They didn't produce any results. And for this past year, in 2023, gravimetric sampling occurred every 2 weeks in all 3 fields. And there is also no existing study that has performed extensive gravimetric sampling like this. So how our gravimetric sampling works: everywhere you see a dot, we dug a hole, and we measured 6 soil depths and determined the soil water content of them. This was done by drying them in an oven at 105 degrees for a minimum of 24 hours. So going back to our overall process, earlier we calculated the N naught through the Desilets equation. The next step was to plot N naught against BWE to gather eta, which is the ratio of intercept to slope, and an average N naught value, or our intercept, and put this back through the Desilets equation, except instead of solving for N naught, we're solving for N now this time. I think my mouse can see there. Then we take this ratio of our N estimate, put through the Desilets equation again, and compare it to our first original input of neutron counts up here to render a vegetation correction factor. So first we had to plot the N naught versus BWE relationship. Looking at this, it is a linear relationship. This has occurred for all sites, for all fields, and all years. So it's pretty clear that this is a linear relationship. There are some studies that have proposed nonlinear, but we have found that a linear fit works best. And note of the R squared value. For most studies, an R squared of 0.47 is a terrible correlation. But for a study like this, it is actually a very good correlation, indicating that a relationship does exist in this nature. And this was seen for a gravimetric sampling as well. Everywhere you see a point. 1 point represents about 108 soil samples on here, which is hard for me to fathom after digging all the soil samples. But we inherently see here, the R-squared has improved greatly, but it is still a linear relationship. And this plots all weighting methods used and all years of TDR data. And we see again that for all three fields, this is CSP2 and this is CSP3, there is a linear relationship that exists. Yeah. So now we look at the intercepts and slopes because in a perfect world, these will all be constant year to year. But what we see is that generally they do follow a similar trend with some outliers here and there. But what this means is that we can use one N naught value for all row crops. If we look at the slopes, which represents the rate of change in N naught with BWE being present on the surface, for the most part, they're clustered rather well again, with the exception of 2020, which is my problem child. The sensor was flipping on and off every 2 weeks, and due to COVID, no one could go out and service the sensor. So that one is treated as an outlier and excluded from all further analyses. So now our key article of interest in those lines is what is the slope of that line? What is the rate of change in N naught with BWE, which is our slope to intercept ratio presented here for our 3 gravimetric weighting methods, and then down here for our TDR method. So now the next question emerges, can we use TDR in place of gravimetric? Because TDR is much easier, the data is easier to collect, and you can have many more points. So the TDR, where you can have 300 samples, we were only able to do 8 really for the gravimetric. And what we see here is that for all 3 fields, this is CSP1, CSP2, and CSP3, they all follow a similar trend. They all follow a downward slope throughout the year. And from this, it is reasonable to assume that we can use TDR in place of gravimetric. While the slopes are not exactly the same, this could be influenced by factors. In the case of CSP2 for the gravimetric, we have 2 clusters of data because we didn't sample for 2 weeks. And while you're out there, you will know that you can hear the corn growing. So if we miss that middle area, that a week in there, we miss that whole middle area. So while we might not be able to say, if you really want good data on July 15th, you have to go out and sample on July 15th. But if you want a seasonal average, TDR is reliable to use. Looking again here at our table of intercepts and slope for gravimetric, they are very similar to TDR, and we can reasonably assume a seasonal average can be produced from TDR sensors. Next is our biomass correction factor, FB. Note that this is basically the inverse of our N naught plot from before. So what we have here, the commonly accepted correction factor was proposed by Batts et al. In 2015, which is this blue line here. And this is the results performed from this study. Now what we note for CSP1 here and CSP2 here is that our lines at least double the Batts et al. Line. But then the interesting thing is that CSP3 is on par with Batts. And there we have a few potential reasons for this that are going to be presented in a minute. But the value we're interested here in the FB line is this average value. So we note that CRS 2000 sensor has a 1.3 average, while CRS 1000 is a 0.6 average, meaning the 2000 nearly doubles the 1000, which would make sense because the 2000 counts double the neutrons as the 1000. And we decided that instead of having a sensor-specific correction, it made more sense to have an overall correction. So we just averaged these 2 values to gather our correction equation of FB. So now the question emerges, we have this Batts correction factor that has been commonly accepted, and we have the one that's been presented here. Why are they different? So the first problem is that this study is on crops while that is in a forest. Inherently looking at a tree and looking at a soybean, they are 2 very different things. And not only are they different to look at, the problem is the way hydrogen is distributed within them. In corn and soybean crops, it is seen as a layer of consistent water across the surface, whereas in forests, it occurs in random clumps of hydrogen all over the place. So the neutrons do not travel as predictably in a forest as they do in regards to travel time or path length for a forest versus a crop. Another reason could be unaccounted hydrogen pools. If you've ever walked in a corn crop at 6 a.m., you will be soaked by the time you walk out head to toe. And the problem is there is no way to measure this water being intercepted by rain or just dew sitting on the plant. And remember, the sensor doesn't see just the water we want to see. It sees all hydrogen pools. So this unaccounted pool of hydrogen could be significant. The effects of microclimate, because our sensors rely on relative humidity and pressure, remember. And anyone that's walked in these two environments will tell you they are very different environments. So while our sensors are more in these clear-cut kind of areas, a lot of the measurements are occurring in these dense row crop areas, which changes our humidity and pressure within the row. And again, different sensor types. The 2000 counts twice as many as the 1000. And simply noisy data, because the TDR, we averaged over the day, but within a day, you can range from 10% water to 50% water. So now if we look at these factors and how they contribute to our difference in the plot, we have this geometry effects are going to have a huge influence on this difference, as well as the unaccounted pool of hydrogen. The sensor type of microclimate shouldn't have as big an impact, but noisy data will have a significant impact. So what can we conclude here? Does a single BWE to N0 correction factor exist? And results suggest that yes, for row crops that have most of their biomass above ground, they can be used as a single factor. So with these plots, this year was corn, the second year was soybean, third was corn, fourth was soybean. But the thing is, you can't tell that looking at this plot. You can't say 1 year was corn or one year was soybean. They all look about the same. So this means that a single correction factor can be used for all primarily above ground biomass row crops. This may not be the same for sugar beets or potatoes or something with more below ground biomass, but that needs further studies to confirm. And recall, this was the correction factor determined in this study. So what is the nature of this relationship? Overwhelmingly, it is linear. Every site, every method has had a linear relationship existing. And can TDR sensors be used as a reliable proxy in place of gravimetric sampling? Yes, over the course of a season, it can. If you're looking for June 15th, maybe not, but over the course of a season, it can. So where does this fit in the big picture of the world? With the development of FB here and in the future, the Cosmic Ray Neutron Sensor can be used more easily, be more accurate, become accessible to the average producer beyond research contexts, and conserve water by minimizing our losses to evapotranspiration, deep percolation, and surface runoff, which can assist in global water conservation. Here's some references, and I'd like to thank Dr. Trenton Franz, Aaron Day, Andy Suker, Brian Wardlow, Sophia Becker, Bidon Demir, and Callie Collins. Anyone have questions about that? Yeah.
So, something that I particularly look at is like biomass variation, that's, like the radiant value variations between irrigated and non-irrigated field, and the agreed upon like causality of like the measured radiant value being different, but the crop being the same is, paper pressure deficit. So do you think that like adding in some sort of correction factor, like in the vision we were talking about, or like maybe one of those reasons that this change is occurring, other than the double the amount of neutrons it reflects, it's also because of the relative BPD difference between CSP2 and CSP3. I have two other questions that are not really at the beginning. One is, do you think like the upcoming P-band satellite called Snoopy might be more helpful towards your work? It's piercing around 0.5 meter into the ground, and don't remember the last question. So, but yeah, great presentation too.
Thank you. We actually just discussed this morning, all of those things in our meeting this morning. But so BPD, we actually have looked into using BPD as a metric instead of BWE, because BWE takes a long time to go out and measure and weigh and collect. The problem with BPD is that when you have a rapidly growing crop like corn or soybeans, it worked good for the second half of the second half of the year after senescence occurs. But the problem is when you're accumulating all that wet biomass, it isn't reliable. It just kind of gives you a flat line. And then, yeah, so Snoopy is actually very interesting. So one of the problems with CRNS versus remote sensing is that remote sensing is lacking field validation right now, like it's lacking the real measurements. And the problem we see with Snoopy right now is that how are we going to get the real field measurements versus their measurements? And then the influence of, like one of the influences with the cosmic ray sensor is it doesn't sense as deep if there is more water present. So that's kind of the other issue with Snoopy is no one really knows, is it going to still penetrate that deep if it's wet?
And my last question, what's the batch, I should probably know this for interest, but what's the batch of the cosmic ray sensor?
So it has an intermediate scale from about 150, so radial, it goes about 150 meters to 330 meters, and it ranges based on your atmospheric effects, your water vapor, things like that. And then it measures up to, some people say 70 centimeters is the deepest you can get, but that is if you have bone dry soil, like no water present. Whereas if you have quite a bit of water present, a realistic range is from 10 to 20 centimeters. So it captures most of your root zone. And like one of the benefits in this study over the TDR sensors is the TDR sensors are installed at 10 centimeters and 25 centimeters, where the you're going to miss that top layer where most of your action is occurring, that first 10 centimeters, whereas the CRNS will capture that variation.
Any other question? All right, thank you, Tanessa. All right, so let's also remember to sign up if you want to present. You still have spaces for people who want to. All right, thank you.
Thank you. I tried to I tried to chew it down. I'm like, what do I got without like missing something important? You did good. You sounded very